Well, good afternoon, everybody. We might be relatively small in number, but I have no doubt we're high in quality. Again, this was originally intended as part of a series, but I was trapped in Cambridge by the gales that we had a month or so ago. Nobody could leave Cambridge. Terrible thing to happen to people. So I'm going to be talking about plant disease, and I want to take you through some of the developing epidemics that we're really concerned with of natural and agricultural systems, and get you, I hope, to have a feel for how we solve some of the problems that these present to us in thinking about how to deploy control and how to be better prepared. So what are some of these global and regional threats? Many of you will remember Dutch elm disease, really changed the countryside. Some of the others that I have on here will be rather less familiar. Cassava mosaic disease is a devastating disease of a staple crop in Africa. This one, cleverly called Rhizomania, root madness, actually threatened the sugar beet crop in a large part of Western Europe and including the UK. That is caused by a fungus, a virus rather, that is transmitted by a fungal-like organism. Soil-borne, this is a fungus caused by a fungus, this is caused by a virus. So we're dealing with a range of microorganisms, and I want to mention Cassava brown streak. That, together with Cassava mosaic virus, are really threatening the livelihoods and survival of large numbers of people in East Africa. And there's a fear that this Cassava brown streak, a viral disease transmitted by an insect, could spread to West Africa, where 160 million people in Nigeria, of the order of 100 million, reliant upon staple such as cassava. Really urgent work is needed to predict where it's going to happen, to do something about it, and then to have longer control. I'll also be talking about sudden oak death, a very different sort of threat to natural environment in California and also in the UK. Citrus canker, which is threatening the production of citrus disease, and there is another one to go with that. Citrus greening, the wonderful title Huang Long Bing, which means little green dragon because it produces those sorts of symptoms on the leaves. Both of those are bacteria. And of course there are the usual problems of fungicide, pesticide, and associated antibiotic resistance. So the tools that we have are themselves being threatened. Ash dieback has hit the news within the last year. That's what it looks like after it's been through Poland. There are some incipient infections in East Anglia affecting crowns of trees and younger trees also. And I'll talk a little bit more about that. Other examples, Phytophthora dieback, Jarrah disease, as it was known, because it affected the Jarrah vegetation, particularly in Western Australia, but throughout Western, Southern, and Eastern Australia. These are the regions here where you get devastating effects in the natural environment. That is a Phytophthora. For those of you that recall the potato blight, that is also a Phytophthora. It's a fungal-like organism. Sudden oak death, as I mentioned to you, threatening large areas of California, and we'll come back to explore that shortly. And also the death of larch. A little hesitation there because I had hoped to have a slightly different slide to show you here. And this spreading, here we see the spread happening over time. That's the prediction, as you see time changing here, of where this rather important disease killing Japanese larch that bulks up on rhododendron is likely to have its major effects in Wales and predominantly in the west of Great Britain. But it isn't only diseases of natural systems. We've got diseases, the wheat rusts, for example, and there are two sorts here, very cleverly called, one is called yellow rust or stripe rust because it's yellow and stripy. You see green, a little green dragon, yellows, very profound nomenclature. This is yellow rust. There is a new strain of that spreading worldwide, and I'll come back to this one, which is called stem rust. It's a rust. These are fungi. It occurs on the stem, and it's, again, cleverly called stem rust. Puccinia graminis, Puccinia striiformis, the Latin binomials. These are really worrying because they can spread long distances and they can spread very rapidly. And here is an example with a paper a while ago from some colleagues of mine ruminating about stem rust. Would it destroy the world's wheat crop? These are the areas in which wheat is grown. You can see particularly important up here, also in the grain basket in the Indian subcontinent, North America, South America. Wheat worldwide, it transpires, has been successfully controlled by genetic means. This is straightforward plant breeding for very long periods by a particular resistance gene. The problem is that all the wheat worldwide has more or less the same few resistance genes, and there is a new strain that has developed of this rust pathogen in Uganda and is spreading out of Uganda, moved through into Kenya, into Ethiopia, and up. It's now in Iran, and the real major fear is it going to get into, as I say, the Indian subcontinent, because there would not be sufficient pesticide to use to control the disease there. So I'll come back to that a little bit later. And there again is Cassava brown streak, which is this devastating disease. So what are the challenges? Early detection. If a disease has spread very far before you realize it's present, then it's going to be a lot harder to control. So one wants to predict, first of all have a method for early detection. Secondly, to predict the likely disease spread and the impact. So imagine you do have a new pathogen coming in, causing a disease, but you're wondering, is this important or not? And you ask two questions: how fast is it going to spread, and what effect is it going to have on the hosts? You have then to predict whether or not it's going to move fast, it is going to have a big effect, and then you think, what sort of mechanisms have I got to control this? Do I have any pesticides? Can I remove susceptible hosts in such a way that I form a cordon sanitaire around where this is happening? And if so, how do I optimize the control strategies? We can formalize these into some epidemiological questions, which are, why do some diseases take off and others don't? That's about invasion. Because some do arrive and nothing much happens. Why do some races, that's particular strains, and diseases die out? Some coexist. So you get a new strain coming in or a new race, you've got an old race which is ticking over there. Do they coexist? Does one replace the other or not? And does one come to dominate? Variability. How do we translate the variability that we see into risk? And I'll come back to that shortly. And scale. New infections occur at the level of the individual. So we feel ill occasionally, hopefully only occasionally. And the question is, how many other people are feeling ill? How rapidly is this spreading through the population? How severe is it? And also, how do we control? So I think I show you one equation in this lecture, but the equation is not important. It is really, what are we attempting to do here? We're taking a complicated system that we know relatively little about, and we're attempting to say, these are really the mechanisms by which I think this system is working, keeping it as simple as possible, and using that to predict what's going to happen. I mentioned Phytophthora, potato blight, Phytophthora infestans to you. And if I ask a biologist, to outline the life cycle of Phytophthora infestans and then to list what's important. Before I can stop my biological colleagues, they will fill 3 sides of fine notepaper with everything that should be in the model. That is not a good way to start, because if you try to produce a mathematical model that you will then use to predict what's going to happen, with something as complicated as this, firstly, it'll take a long time. Secondly, it'll be very difficult to test it to see whether or not you've really captured what you ought to have done. And thirdly, it'll be redundant because you've got detail in there that is not essential. So what do we do instead? Well, we think and follow the lessons, in fact, of medical epidemiology. And let's just think a plant is susceptible, then it becomes infected, and then it's removed in one way or another. The removal means it's either recovered or it is dead. It's rather unfortunate that those two are pushed together in a single compartment, but from an epidemiological point, it means that these individuals here, let me take the frightening bit away, that these individuals here are no longer part of the epidemic. They're not capable of spreading the epidemic. So that's what really matters in terms of spread. And it's these individuals, the infected and hence the infectious individuals, and this beta S for secondary infection is showing cycles of infection here. As well as the cycles of infection, so think about an epidemic spreading in the part of London that you live and work in. It'll cycle in there, but you'll also have people coming in from outside who may also be carrying some infection. And that's shown here with the external inoculum coming in. 2 sources. That's the usual epidemic. This can trigger it off, and when it subsides, it could trigger it again by something else coming in. It's possible to write an equation for this, and I really don't want to dwell on the equation because I want to go from there to thinking about we can adapt this in various ways. Other numbers of plants grow, so the number of susceptibles increase over time. They also die, and we can allow for those. Of more importance is to think again about susceptibles, infecteds, and removals or removeds. Focus on the middle graph here. We have time, and we have the number of infected scaled here, where 50 is a maximum. And I want to make a couple of points in this. The first is that we have an epidemic here which is just about taking off. There is a concept known as the R0 concept here, that on average, if one individual infects one or just slightly more than one individual, the epidemic will take off. If on average an infected individual infects less than one individual, averaged, then the epidemic will fade out. It's quite a simple concept, quite a sophisticated mathematics sometimes underneath it. We're looking at an epidemic that, in this case here, that is just at that stage. It's just about to take off. And if we use what is called a deterministic model, we have that line here. Let me just put the explanation in here. That's the deterministic model. The number of infecteds will increase, but then the epidemic will fade out. Very familiar with to many of us. And the mathematics underlying this is the sort of mathematics that we learned in school, that you have an equation. For whatever you put in on the right-hand side of the equation, you always get the right answer out on the left-hand side of the equation. Would you like some? Okay, but if in need, I haven't touched it. So that's the deterministic model. But in fact, life is not like, and death is not like that. It's not deterministic. Chance plays a part. And so what we find is if we use a so-called stochastic model, which is a model that allows for the inherent variability in the system, we have something where many of the epidemics, this is looking now at many epidemics repeatedly, under the same conditions, many fade out. Those that do take off, this region here, you can see you can have as few, at the maximum, as few as that number of individuals infected, but as many as that. And so for planning purposes, one really needs to understand that sort of distribution rather than assuming that we have something as simple as that. That's the difference, therefore, between a deterministic and a stochastic model. So what about estimating parameters from an emerging epidemic? So a parameter now is the tuning that we're going to put into the mathematical equation, which is saying, is it taking off or not? And a simple parameter is a transmission rate. So again, I feel healthy, but if I were infected, there is a risk that I might transmit the infection to Professor Cox. And that would be measured by a transmission rate. And if I moved further away, there is also a dispersal kernel. So what's the chance if I sneezed? I do feel healthy. If I sneezed, if I'm further away, then the probability of transmission decreases. And I'm going to use an example here of something called Asiatic citrus canker, but I could have chosen one of many. It's caused by a bacterium. I've already introduced it to you. It's transmitted by rain splash, rainstorms, can be exacerbated by this leaf miner here, and it is in danger of eliminating citrus production in a number of parts of the world. There was an outbreak back in the 90s. There have been others in Florida. Starting in 95, this is the region in Miami, and the new regions infected in each year are in red, and you can see the epidemic progressing here. A very expensive eradication program. 11 million commercial trees, 1 million dooryard trees are the back. Why am I apologizing to the microphone for knocking it? I'm apologizing to you for the noise. Dooryard trees are backyard trees. So we want to understand, how is this spreading? Can we predict the spread? And importantly, can we predict how control would work? So we take a series of snapshots of disease over time. And in particular, if we home in, we're going to go into a particular region here. And my colleagues in USDA actually went into these regions that's shown here: Broward County, north of Miami, Dade County. And in successive months, they measured, looked at every tree and scored the tree for healthy or infected. And that's what's showing you here is 180 days, you had that number infected in D1 here, 750 days. And that's what the disease progress looked like in these different regions. Different rates of progress, intriguingly. So we have monthly snapshots. And I've left a picture of one of my collaborators, Alex Cook from Singapore, in here. What we do is take these successive snapshots over time. That's showing you the green are the healthy ones, and the red, in this case, are the new infecteds. The black are the previously infecteds. Month 0, month 1, month 2, 3, 4, 5, 6, 7. You can see the epidemic developing. And we use an epidemiological model, a lot of statistical methodology, a lot of computing power, and a lot of toil to produce something that produces graphs. Now, what do the graphs tell us? They tell us about the dispersal kernel. That is, as you move away the infected from the susceptible, how does the probability of transmission fall off with distance? That's what that alpha is about there. The transmission rate is, if the inoculum reaches an individual, what's the chance that it infects? And that we also have the external infection here. What's important about this is, if you simply use the first few months to estimate these parameters, you get something which is saying, we have a big range here. We don't know what this transmission rate is. It could be as small as that. It could be as big as that. And you can think, again, from the stochastic models that I indicated, that's going to give you a huge variation It's a bit analogous like many human diseases, prion diseases, for example, where we don't know just how infectious they're likely to be, and so we have very big ranges. Moreover, as time goes by, we see that the distribution here, that is the range for what that transmission rate might be. You start with a very wide range, and then as you add more information, the range comes in. But we also found that the transmission rate was changing over time, so it was becoming less because there were drought conditions coming through. So I wanted to give you a little feel for, here's an emerging epidemic. You're trying to work out, much as we're doing now with a range of diseases, how important is it? Can you get some sort of signature out of those data? And then you want to predict ahead. So here is time. Here is level of infection. The red, are they still appearing red or orange? The reddish orange on here are saying that's where we know disease to be at present. That's where it turned out that it went, but at this time here, we don't know that. We're predicting, and that's what we're predicting here. As we move on and we get a little bit more information, you see the prediction a lot better, and as we move further on still, you can see that you're honing down what the variability or the uncertainty is likely to be. And this becomes really important because when are you going to make a big decision? And I work a lot with policymakers in ministries and in government departments in the US, the UK, Brazil, and elsewhere, where you have to make a decision at an early stage. Are we going to do something about this or not? But look at the range that you're having to deal with. So there is a trade-off as to how long you wait before you do something. But if you wait too long, you're going to make it harder to deal with it. But it can be done. So let me now move to another theme that I'd like to explore. If this were the usual sort of audience that I don't want to imply that you're an unusual audience, but something I would often do at this stage is just try to gauge the interest quotient. How are you feeling? I can usually tell that from the faces, yes. We'll continue. Okay. Now let's think about how we might go about thinking in more detail about an invading pathogen. And what you really need to know, so the pathogen, of course, I'm sure this audience knows, but the pathogen is the organism that causes the disease. One needs to think about the host distribution. How are the hosts arranged in space? And this is important in human disease, where we're thinking sometimes it's about spread through villages, spread through primary schools, secondary schools, where you get a lot of movements. In plant disease, it's again the contiguity or otherwise and the gaps between the susceptible hosts in one region to another. The environmental conditions, how important are they? Temperature, relative humidity, and so on. And also, I've given you a feeling, I hope, for some epidemiological models underneath these. And these are used to understand and predict disease. There are many examples that I could have chosen, indeed, from work that we've done and others, but I'm choosing sudden oak death that I mentioned to you before. It attacks 100, at least 100 species of hosts, but it is particularly devastating on some of the tan oaks and coastline oaks, which are different genera, in California. There you can see some of it on the tan oaks spreading. Bulks up on, in this country it bulks up on rhododendron. In California it bulks up on bay laurel. And those of you who've been on the West Coast, there's a lot of bay laurel there. We needed to look at, for USDA, United States Department of Agriculture, the spread of this disease. And we've resolved the vegetation to 250 by 250 meters. We need to think about transmission, which depends on weather. Dispersal occurs by short and long distance movements. And there are some technical details about estimation here, but let me take you through. That's a vegetation map of California, remarkably well resolved, but that's not a lot of use to us at present. We need to think about that vegetation map in terms of where are the susceptible hosts. So we take this complex map, we think about the species that can be infected and the density with which they occur, and we scale this. It's on an odd scale here, but it's essentially 0 to 1. The darker regions here indicate there is a high density of the more susceptible hosts, down to the very pale green, going to white, where there are very few susceptible hosts. So we have a feeling now, and the equivalent would be looking at population densities of human population if we were thinking about a human disease. We need to think about weather, and it transpires that weekly rainfall, weekly temperature are important for the spread of disease. But what we want to do is to think, in any given year, what do they look like? How does that change? How does that change? And how do they combine to produce favorable conditions that turn on or turn off the chance of disease spreading? And if we just run a little simulation here from the real data, keep an eye on how this changes and how dark it goes. When it's dark, it favors a spread of infection. As we run through time, you see that it's not that often that you get the dark pink occurring, and this becomes very important in understanding the role of the weather in the transmission. We can put these together, and we know that the infection was introduced here in the Bay Area at 3 small sites, and produce a risk map which is running through from when we believe the infection was introduced, early 90s, right through to 2030 or so we were looking at. And as we run this, you'll see the infection risk changes according to the time here. The green is the susceptibility of the vegetation. The blue going up to red is the density changing over time. And as we run the model, watch what happens up here particularly. We shall see that in fact the disease, the probability, this is us predicting what's going to happen, and we saw this changing here as we went through time. This is going out to 2030, which is a sensible time to go for some of these systems. One can predict the spread, and note that even though we introduced the disease in here, it's still spread by jumps. We just put very simple rules in for how this might move. And we pick up something up here. And if we compare that with the known data at the time, indeed, we saw there's a lot of infection down here in the Bay Area, moving up. But then in this Humboldt region here, particularly suitable contiguity of the hosts, high degree of susceptibility, favorable environmental conditions, and a hot spot there. So the model is actually doing very well. But I would show you one that does very well, wouldn't I? I have plenty of others. Returning to that theme of uncertainty, when dealing with the policymaker or the regulator or the landowner, what we can say is we can tell you the chance that your region is going to be infected. And here we need to distinguish between running a model many times over and averaging out. So you recall that I showed you that graph of the uncertainty, the stochastic model, the shading. We're running it many times, and the intensity of the shading tells us what is the average expectation for disease in that site. We need to contrast that with that risk over many runs and compare it with a single run, because in truth, when you have an epidemic, it is that single epidemic that's running. It's just ahead of time. We don't know which of the many galaxy of different epidemic transition sequences that could actually be happening. So let's just go through this again. We have I'm going to show you a single run on the right-hand side. And we do that once, then we do it again, then we do it 10,000 times and average those and put them here because that's the best bet in order to be able to predict, does it get over into the Sierras, for example. And if we compare these two, you'll see some differences. So average on the left, one particular, and note how slow one particular run might be to go, and that's reflecting a particular epidemic. And as we move through, you see the particular single run is quite slow, but if we average these, and we don't know yet what's going to happen in 2028 until we get there, there is an important distinction to be made between the prediction based upon the average of many runs and then the particular run itself that you will be involved with. And you update that according to the information that you have got. So that's using a risk map, and we can distinguish this from a hazard map. Let me see if I can explain this. The risk map is saying if we look from 2000 up to 2030, what's the chance that we get the disease? And this is based on many runs over in the Sierras, not very high. What's the chance that it spreads down here in the Big Sur? Possible, but it doesn't spread very far south. And this helps us to work out the probability that you get infection in these different regions. And also, what's the mean infected area? So we've now taken, you can see the NW refers to the Northwest, Sierra Nevadas, Central West, Southwest. That's the probability that those are the probabilities we get the disease in those different sites. And also, the colors here correspond with the colors up there. What's the likely area of severity? That's a risk map because it's saying where it's likely to go. We distinguish that from a hazard map, which is actually saying not what's the probability that a disease gets into a particular region, but if it gets there, how severe will it be? It may seem like a subtle distinction, but it becomes very important if you are a Secretary of State and you're trying to work out where you should deploy resources in order to see if the disease is there, to do something about it. And we can see that that's a hazard map. Compare that with the risk map. Difference because we're saying if the disease were to be introduced up here, deliberately or otherwise, it would spread very rapidly. And so then you think about the economics of the situation. How important is that region? How much resource will you put into detecting the disease there? So we've distinguished some risk and hazard maps. That was a little bit more complicated. How's the audience feedback at this stage? Okay, thank you. Let's think now about multiple methods for invasion. And here I want to distinguish, I'll be turning to ash dieback, which was very much in the news this time last year, trade imports versus aerial transmission. So thinking about ash dieback, we have some pictures here from the Forestry Commission. You get death at the top of the tree. Here is a sapling, however, with different set of symptoms. We have two types of death occurring. One is in the so-called wider environment, but is essentially in mature trees. And some of you may well have seen ash trees with that dying and stag head there. And the other is in, usually the saplings will be rather shorter than this, where we've imported saplings from the continent. These are sites at which the disease has been detected. Up until early this year, we have newer data. And what you see, of course, is that most of the disease in the mature trees is on the east coast. The saplings, of course, have been imported throughout Great Britain, and I haven't put the data on for Northern Ireland and the Republic, but these are coming through. The question is then, how important are these potential different sources of infection. It's not for the first time do we in Britain turn our eyes on the continent and think the problem must have come from the continent. Okay? And if we trace what we know about the fungus Cholera, it's a fungus called Cholera. It first occurred in Poland in about 1992, and as we step through the blues, coming through the yellows to the reds, you can see the progression here as recorded throughout Western Europe. There seems that there's a high probability, therefore, that if this is aerially transmitted, but note too it's also transmitted by the trade, that there is a lot of potential inoculum. And Professor Cox said, plants are easy to paraphrase because they don't move, but of course the pathogens move. And although the plants are static, you've got spores moving or viruses being moved by insects. I have paraphrased what you said. So we're now asking the question, what's the likelihood that aerial transmission could have spread the pathogen from the continent onto this suspiciously asymmetric region here? It's one thing to be suspicious, it's another thing actually to have some evidence. And let me just go back for a moment. Remember too that the prevailing wind direction, notwithstanding the picture I'm about to show you, is in that direction. Definitely the prevailing wind is in that direction. So we used a model from the Met Office that's been used to track pollutants, though this is just showing the effect of water vapor. But you can see the wind effect here, and that's essentially what we're looking at, not implying that there is pollutant coming out of these. So what we do is think of all the many sites, and I'm just showing you 3 out of several thousand sites. This is computationally very intensive. I mean, the computer feels hot when it's running, and it's a big computer. So for each and every day over a 4-year period, we say, was there infection present in a whole set of regions here? And what were the conditions like for wind? And you can see in this particular 2nd of June, I've just shown 3 trajectories from my own lab here. That's where France would have spread from. This particular one in Germany was going the other way. And we have an interesting one up here in Norway. But you take all of those over, we looked at a 4-year period because that was what we thought was the most likely region. Data very kindly supplied by the Met Office. What we were able to do was to identify which regions were the biggest donors of potential infection from these spores. And it's difficult to read that, but the size of the dot indicates, if it's bigger, it is more likely, based upon the historic weather data, to have been a donor. What's striking in this is that you can get right over here into Germany, and that's still, because of the weather patterns, likely to be a big donor. As this spreads further south, these regions will, of course, become more probable donors. But this in itself was a major revelation. Also, it was it involved some novel computation. So as far as I was concerned, it had everything going for it, because it was new mathematics, new computing, and a useful result. Not necessarily in that order. And what we see is that we then consider you'd be familiar with these sorts of scales now, from low to high, the relative deposition of spores in the British Isles from the region here in the continent. And of course there is very close correspondence with those infected sites in the wider time. There's a lot more statistical analysis that has been done on this to compare the magnitude or the force of infection from those sites that were introduced on infected sites on saplings. They do have an effect, but not nearly as great as that aerial transmission. And the bad news of that is, of course, that there will be continued aerial transmission from large regions infected on the continent. So very strong agreement, as I indicated, and a good predictor of arrival. We've taken this idea a little bit further in terms of, you recall that I mentioned wheat stem rest to you. This was the one that was moving out of Uganda. It is spreading north into Iran and also down into South Africa. It's called UG99. The races are called UG99. Uganda, 1999, it was first identified. What we're very concerned about, as I indicated before, however, is the threat to wheat worldwide, and in particular, in collaboration with the Gates Foundation, the threat within Africa where wheat is relatively much less important than in this region here, where wheat in Pakistan through in northern India is a very, very important staple crop. And so we're doing the same thing again of tracking where have we got infection. That's what it looks like early on. This picture was sent to me last Friday when I got an urgent email from some collaborators in Ethiopia involved with our Gates Foundation project. And the email began urgent. They had a new race of UG99, so it's UG99, but they discovered it in 2 regions in Ethiopia where it had not been present before, first point. Secondly, it was much more severe than had been seen previously. And the urgency since then, over this last week, has been to look at where that is likely to spread, given that it is there. Is it likely to come up here in significant amounts? In fact, the evidence, these are just single snapshots for particular days, but the evidence at present is that it's more likely to be spreading in this direction during the October and November weather. The critical feature is what's going to happen in this December and January period. So that's where you develop the toolbox. You then get some emergency situations like this, where you're attempting to predict where it's going to go next so that you could get some pesticide there in time to be able to slow the epidemic. But you're not going to get any yield from that. So again, using the meteorological dispersal model in collaboration with our colleagues at the Met Office, integrating lots of sources of infection. But note that although this shows potential here, there's a very low probability down there. It's only a high probability in that region for that particular example. So let me give you one more example. I'm thinking about control. What I've done up to now, I hope, is given you a feel for the heterogeneity of the spread of disease. It's controlled in part, and largely, by the distribution of the host. It's also affected by the distribution of the varying weather that you have at different scales. And it's characterized by those parameters, the epidemiological parameters that I mentioned, the probability of transmission, sorry that I keep insisting upon just wanting to infect one individual here, and also that notion of how far away is the infected from the susceptible, those sorts of parameters. So there is an inherent scale of the epidemic that is influenced by the pathogen, by the host, and by the environment. And if we're going to control disease effectively and efficiently, we need to be able to match the scale of control with the inherent scale of the epidemic. And we often don't quite know what that inherent scale of the epidemic is. So I'm going to give you an example, an older example, where we first developed this concept. And it goes back to Rhizomania. That was the viral infection that is transmitted through the soil from sugar beet crop to sugar beet crop, from plant to plant within the crops, by movement of, particularly of soil, farm machinery. So you take cultivators in, there we go again, and cultivators move from one farm to another. Now at the time, the government proposed that there should be what effectively is a field scale scheme for control. If you see the symptoms of disease, and the symptoms, if you pick the plants up, you'll see it like this, but you tend to get a yellowing in the field. And then you look at your crop. If you see the symptoms on your field, there would then be a test and you would find that you had the inoculum, that is, you have the spores present. And you could no longer grow sugar beet on that field. But if you think about this epidemiologically, you think, if I've got symptoms and I've got enough spores here to be readily detected in whatever molecular or other method is used, chances are I've been exporting that to my other fields and to my neighbours. So what we, with the sugar beet industry, discussed was a farm scale scheme, which is saying if my neighbour, or indeed, so here am I, if my neighbour or my next nearest neighbour with whom we might share machinery or the same contractors, if they have the disease, maybe I should think I've got it too. So what I could do in this case is say, let's just do it with my nearest neighbour. Here am I, here's my neighbour's got the disease. I then think I must have it, so I sell my sugar beet quota to someone else in the country, and there is until very recently, there was a market for this. Growers wanted to do this. So I sell it to somewhere in the country where they don't have the disease as far as we know. And let's run this again with a lot of epidemiological modelling underneath it and run it through time. Colour scheme leaves a little bit to be desired here, but the white region is where you can grow sugar beet. No sugar beet really grown in these regions here. And we'll run through time. Red is bad, green is good. And we're going over a number of years here. We see that the default scheme that was very contentious with the EU because it involved restricting imports and movements, that's this one, compares very unfavourably with the farm scale scheme. Why? Because we're failing to match the scale. We're working on a field scale, but the spread is on a farm scale, several farms. We're failing to match the scale of control with the inherent scale of the epidemic. And repeatedly, we're now looking at how do we characterize that heterogeneity, that uncertainty that you've got in an emerging epidemic. And it's the same with livestock disease. It was a big question with foot and mouth disease. Did we overkill in order to bring the epidemic under control? And that links, I won't have time to go into it, but that links naturally then to economics. Some time ago I was invited to Washington, D.C. to a meeting of economists who were modelling the spread of disease, human disease principally, and they were doing some very good work, but they didn't have any real biology in their understanding of the system. Nor, for that matter, did we have much economics in our models. So since then, we've been putting the two together. Also, if you ask yourself, who does a government listen to first, economists or natural scientists? A nanosecond should give us the answer. But it becomes very important, actually, to bring those disciplines together and say, if there's a constraint over how much pesticide and fungicide I have, then how should I deploy that optimally? Supposing I'm thinking about two countries, different levels of disease in each, I don't have enough drug or pesticide to go around. Do I preferentially try to reduce the infection in the more infected country, or do I, if I'm an international agency, or do I tackle the easier one? It transpires that for some diseases, to go for controlling preferentially the disease in the more infected country is the worst possible thing you can do. It gets out of control. What you should do is concentrate for particular diseases on the lower level of infection, and then whatever's left over you use for this, and we're assuming that there's some movement between the two countries. So by putting those two aspects together, epidemiological insight, economic insight, some real facts, one can get some counterintuitive answers out over where you should optimally deploy your effort. And I've paraphrased rather than presented each of the slides on that, but we can tackle it and there is time for some questions. So what have I done? Indicated, I hope, some serious threats to agricultural, horticultural, forest crops and natural vegetation, and different causes. I've shown you some epidemiological models, allowing for uncertainty and, importantly, matching the scale of control with the scale of the epidemic. I've given you a teaser about coupling with economic models. Thank you.